Yes, good evening, Hiral bhai.
Good evening. Good evening. How are you, sir?
Fine, fine, thank you.
Good
Okay.
evening, Devang bhai. Good evening.
Thank you, sir.
Okay. Shall we start, sir?
Yes, sir.
Okay. So, good evening, one and all present over here. Today we have with us Dr. Devang Gandhi. He is the director and technical manager of Pollucon Laboratories that is located in Surat, which is recognized by CPCB, GPCB, FSSAI, FDCA, and ZDHC for textile testing. Mr. Devang Gandhi, Dr. Devang Gandhi is an enviro-legal expert with master degree in law, postgraduate diploma in environmental law, and PhD in chemistry. Sir is an environmental auditor from last 15 years and environmental consultant since past 24 years. Sir is also a government analyst gazetted by CPCB, MOEF, Government of India. Sir has an experience of operating almost all the modern laboratory instruments that are relevant to environment and food testing scope. So over to you, sir. Uh, Looking forward for a knowledge sharing session from your end.
Okay, Hiral bhai, thank you very much. First of all, thank you very much, Pacific School of Engineering, especially Rotliwala sir and Hiral sir, for inviting me for such valuable and informative session. I will try my best to deliver the subject specific speech, and sometimes this is very technical, and if anybody having questions or doubts, we will discuss at the last. Thank you.
Okay, sir.
Sir, my screen is visible to you?
Yes, sir. It
Okay.
is visible, sir.
Okay. Should I start my presentation, sir?
Yes, please.
Yes, yes, yes.
Okay. Thank you very much. And thanks all of you for listening me. Advanced instrumentation for analytical practices in environmental parameters. Today, this session is very vital looking to the laboratory analysis, testing and monitoring. I will try my level best to introduce all the modern instrument techniques, including sampling and analysis. First of all, environmental monitoring and testing. Why it's required? It's a growing need with increasing urbanization. All of you knowing this, industrialization and agriculture activity. The need for precise and accurate environmental monitoring is paramount. The pressure on the natural resources, particularly air, water, soil, is increasing day by day. This necessitates a reliable system of monitoring and data collection to ensure environmental protection and its sustainability. This is the problem, and we should understand this. All the human activities are the primary source of pollutants. This pollution poses a significant threat to human health, ecosystem, and overall well-being of our planet. By monitoring environmental parameters, we can gain insight into the extent of pollution and the effectiveness of mitigation efforts. Uh, the role of instruments. First of all, detecting contaminants or pollutants, whichever we should say, and ultimate protecting the environment. Analytical instruments play a crucial role in detecting and quantifying various contaminants present in air, water, and waste. They enable us to identify the specific pollutant, their concentration, and their potential sources. These instruments are often used in environmental monitoring program listed under Central Pollution Control Board, Gujarat Pollution Control Board, or any regulatory board for implementation of the norms. Protecting the environment is our motto. The data generated by the instrument is essential for informing environmental protection policies, ensuring compliance with applicable regulations, and developing effective pollution control strategies. By understanding the sources and level of contaminants, we can take measure to minimize their impact also. Importance of advanced instrumentation. Why we require advanced instrumentation? I will give you one example. 15 or 20 years ago, we are using spectrophotometer to determine lead in water or air or in sludge, uh, which have some expertise or which have some experience of laboratory. Uh, it's very known. The spectrophotometer analysis is very lengthy, very precise, and having a lot of interference of other chemicals with color also. Before 20 years ago, in India, 90% laboratory and also the government bodies having spectrophotometer, the only instrument to determine lead or other heavy metals. Lead determination is very vital because it's covered under all the environmental rules and regulations. So first of all, there was a spectrophotometer from where we can use to determine the lead. I also tested lead on spectrophotometer. Then the atomic absorption spectrophotometer introduces in India, because all these instruments are having big technical and economical implications. The spectrophotometer is readily available within 1, 2, 3 or 4 lakhs max to max. Atomic absorption spectrophotometer 15 to 20 lakhs. Now we change over from spectrophotometer to atomic absorption spectrophotometer for lead or likewise heavy metals. Atomic absorption spectrophotometer is having its own limitation. It's long process compared to other advanced instruments, and yes, detection level. Now ICP and ICPMS are the answer for latest environmental testing for heavy metals. Spectrophotometer detection limit was in ppm. Atomic absorption spectrophotometer detection limit is in 0.1 ppm for water, and ICP and ICPMS will give results in ppb level. So the compliances of environmental regulations will be very easy, and we can identify all the elements in black and white with higher accuracy and higher trust level. So this is an example why we should require advanced instrument, because day by day regulatory limits are very stringent. If we can discuss, before 20 to before 25 years ago, ppm level was acceptable. Now ppb level or trace level are also is questionable. So looking to the national and international regulatory requirement, we should go for the advanced instrumentation. I will cover numbers of instruments today because sensitivity and accuracy always be there. Modern analytical instruments provide high sensitivity, accuracy, and reliability in measuring environmental parameters. They enable us to detect trace contaminant and accurately quantify their levels, even in complex metrics. Which are complex metrics? We will discuss further during presentation. Advanced techniques. The need for advanced technique is crucial to measure trace contaminant and complex metrics like hazardous waste, wastewater, which are having color and other interference of organics, inorganics. The methods allow us to analyze various chemicals and determine their impact on the environment. Matrices under scrutiny. This technical word are somewhat vague, but we should understand because this presentation having numbers of instruments related to numbers of matrices. First of all, air monitoring analysis with quality is crucial for protecting public health and the environment. Instruments measure pollutants like particulate matter, ozone, VOCs, and other toxic gases. Water is one of the matrices covering wastewater, drinking water, surface water of river, marine, lake, and also the groundwater. Water quality monitoring and analysis is essential for ensuring safe drinking water and protecting aquatic ecosystem. And also it's required to match environmental norms, particularly for pollution control board, central pollution control board, and for each and every manufacturing unit. Instrument analyze contaminants like heavy metals, organics covering pesticides, TOC, and overall COD level. Waste covered municipal solid waste also, sludge of ETP, and residue of manufacturing process or byproduct. Monitoring and analysis of hazardous waste, soil, municipal solid waste, and other industrial waste is vital for proper disposal, and environmental protection is very vital. Looking to the pathways, instrument analyze the composition and potential risk of this material of pollutant, and it's also identify the disposal pathways for safe environment as well as regulatory compliances. In India, we are having TSDF sites, municipal solid waste sites, and incinerator disposal guideline, which are different in a way, looking to the concentration of the pollutant. One example is that if a calorific value will be high or more than 2000, such waste will not be allowed for TSDF or landfill. It should be incineration, or any toxic gases, cyanide, including waste, should be disposed in incinerator or the higher techniques available in India. So the instrumentation detecting such pollutants is very vital now. Moving forward, by utilizing advanced instruments and analytical practices, we can gain a deeper understanding of our environment and implement effective measures to protect it. The data contribute to sustainable development, policy decision, and ultimately a healthier planet for future generation. We can achieve the proper detection level with higher confidence and accuracy using advanced analytical techniques and instruments. These are the instruments which I will show some summary usefulness and the limitations also. Liquid chromatography, mass spectrophotometer. It's required to analyze pesticides, herbicides, and other organics which are having higher boiling points. We will further discuss this boiling point and differentiates the parameter instruments wise. Further slides, gas chromatography, mass spectrophotometry, which is used for trace level of pesticides and trace level of organics. Which organics we should do by GCMSMS, by LCMSMS, by GC ECD, or by routine GC analysis. We will differentiate with the modern techniques how and why it's required. ICP-MS, atomic absorption spectrophotometry, ion chromatography, HPLC, TOC analyzer, CHO elemental analyzer, VOC analyzer, yes, NDIR CO analyzer, and online gas analyzers for gases also. HRMS, which determine dioxin furan also, and I will slightly discuss the solid phase micro extraction SPME and other sample preparation techniques, which is vital for introducing the sample in these all modern instruments. Without proper sample preparation, proper result will not be there if we will use latest or modern instrument. But I will light some parameters why this sample preparation technique is required also, including these modern techniques. First of all, I will show you this is the liquid chromatography mice spectrophotometer known as LC-MSMS. I will go through in summary of this principle. LC-MSMS combine the separation power of liquid chromatography. With what is liquid chromatography, we will further discuss in further slides. With detection and identification of capability of mass spectrophotometer. Why LC-MSMS is required and where LC-MSMS is required in our environmental parameters. I am sticking this my presentation today for looking to the environmental parameters also, because these vital instruments is used further research and development also, food and drug also, but we will discuss today looking to the environmental parameters with the examples. Environmental analysis, LCMSMS is powerful tool for detecting organic analysis wherever trace organic ever required. Nitrogen and pesticides which are having higher boiling point and which is not able to determine by routine GCMS techniques, where LCMSMS is the only option to detect, and yes, detect at lower level. Similar way, antibiotics, pharma drug residue are also determined by LCMSMS in wastewater, soil, sludge, and in air also. Key applications similar way is identified and quantify pharmaceutical residue, pesticides, and other environmental matrices pollutant. An example, 2,4-D. We all know this 2,4-D is pesticides, and it was widely used in our environment in fertilizer, soil, and the fertilizer soil overflow, looking to the leachate of water going through uh, the soil and other layers into the groundwater. Also, 2,4-D manufacturing process emit 2,4-D in air. 2,4-D is a dust particle, and we should measure 2,4-D in dust particle through this LC-MS. And all these parameters having its all dedicated method, dedicated sample preparation method, and dedicated sample analysis methods, which is having a dedicated column also. I will not go through and will not discuss very specific columns, temperature, but overview of the same is vital. So the 2,4-D in water, uh, from before 20 years ago or before 25 years ago in India, we will determine by HPLC. But now the same detection level and limitation of accuracy. 2,4-D is not accurately determined at low level in water, which is vital now because the regulatory level are stringent. Soil as well as air sample in air, 2,4-D detection is very vital because in air dilution of the air is huge. 99% of air and 0.1% of 2,4-D or such other pollutant are there. Sampling is very vital, and after sampling, how to detect this 2,4-D or other parameters which are having very less in quantity, ppb in ppt or in ppt. So in this case, this vital instrument and modern instrument are required today. LC-MS. Wastewater monitoring, LC-MS is essential for monitoring of trace contaminant in wastewater. Further, higher boiling point organics like pharmaceutical residue, pesticides, and yes, antibiotics. We will discuss for antibiotics why it is correlated in our environmental parameters. Surface water analysis, it helps to assess the presence of pollutant in river, lake, and coastal water, ensuring water quality and public health. More or less, all are pesticides overflow. Sediment analysis, this instrument is determine the presence of persistent organic pollutant. We will further discuss which are the persistent organic pollutants and their impact also in summary. Same case with sludge waste as per the hazardous waste rule 2016, which are applicable in India. Also marine water monitoring, marine sediment monitoring, how it can be determined and how this project will implicate and require LC-MS. This is summary: liquid LC-MS, high sensitivity, versatile analysis, and ultimate data interpretation of software. LC-MS can be detect very low level, very low concentration. Also in complex matrices, it will make ideal for trace contaminant. I will show you the 2,4-D where HPLC, the limitation of HPLC, and now we have LC-MS. which is up to ppb level. So water, soil and air all are combination of this useful instrument. This versatile analysis and different techniques using data interpretation. Now it's very easy to identify, quantify such parameters like 2,4-D. Is cover all the pesticides, herbicides and uh, pharmaceutical intermediates also. Now from last 2 to 3 years there is some guideline and norms published by CPCB regarding pharmaceutical intermediate. This is the only instrument which determine the pharmaceutical intermediate in wastewater as well as sludge at trace level, which is regulatory protected by CPCB under wastewater and soil, as well as now other publications are coming in air and from stack emissions also. It's will be very short time. This pharmaceutical emission will be differentiated in air also. Today there is no limit in stack gases or incinerator. Yes, some parameters are covered under factory rules, but not under our environmental rules. Now, for low boiling point, higher boiling point organics, we can differentiate organics in two ways compared to the analytical techniques available with us. One is low boiling point and one is high boiling point. LCMS MSMS is very useful for low boiling point analysis as GCMS, but the limitation is there. So we can use LCMS for higher boiling points, and now gas chromatography mass spectrophotometry GC or GCMS or GCMSMS for low boiling organics are very easily accurately quantified by GC, GCMS and GCMSMS. Why we require GCMSMS? We are having GC or GCMS. We will discuss further the limitation of all these instruments. First of all, we will discuss the principle of GCMSMS in summary. It's a powerful analytical technique combining two complementary methods. In the first step, routine GC, gas chromatography, which separate different volatile compound based on their boiling point and interaction with a stationary phase as column. The separated compound in column then enter the mass spectrometer where they are ionized and fragmented into the characteristics ion, and for which we easily compare to the NIST library, NIST or other library. Structural elucidation. The MSMS techniques allow further identification and quantification of compound by analyzing the fragment pattern of the ion. This process, known as tandem mass spectrophotometry, provide detailed information about the molecular structure and its identity of the compound, offering high specificity, sensitivity, which will identify as well as give confirmation for identified compound. GC MSMS is very easily achieved. VOCs, semi-VOCs, POPs. What is POPs? We'll discuss it. Now, first of all, VOCs like BTEX, as we all know, benzene, toluene, xylene, and other solvents. All are very easily detected in GCMS, where GCMS is required, where unknown sample is there. We are not knowing the characteristics of the sample, and the fingerprint analysis is required. We will discuss what is fingerprint analysis in the further slides, but we will stick today and at this slide for VOCs, including hydrocarbons, aldehyde, ketone, and all the alcohols. These compounds are often associated with air pollution also and industrial emission also. Most of our we are knowing that there are national ambient air quality guideline and norms which is having benzene. Yes, benzene is covered under the VOC, and it will analyze very easily by GC technique. But for unknown air compound, GC MSMS is required, where PPB or PPT level is required, because in our air huge dilution is there, nitrogen, oxygen, and the component of pollutant is very less. So at that time, this GCMS MS technique is required. Identification of VOCs. The technique can also identify semi-VOC also. Which are VOC? Which are semi-VOC? It's on the boiling point analysis and differentiates on the boiling point. Such as pesticides, some pesticides, most of the pesticides having higher boiling points, and most of the solvents are at low boiling point. So this technique of GCMS, LCMSMS, and GC, GCECD, GCTCD, these are the very technical words. Who are in the laboratory, they will know. But I will some example with the detector also. These herbicides, phthalates, which are often found in soil, water, and biological samples also. I have write down the method of preparation test method. We will not go through, but if anybody having some knowledge and wanted to go through some methods, these methods are very useful. So I will not discuss technical methods, technical sample preparation method, sample analysis method. This is the method which is described on this slide. If anybody wants to go into detail, please go through this method. This is a US EPA method. Most of our Indian method are derived from US EPA method. We will discuss what is the use of GCMSMS and which matrices. First of all, air quality monitoring have a vital role of GCMSMS, VOCs and organic pollutants. This data helps to assess air pollution level and guide regulatory body like GPCB, CPCB, and also industry for complying these statutory limits, which is uh, uh, published by CPCB, GPCB, or any other regulatory body like factory rules, factory rules under NIOSH, OSHA, and other manufacturing facility, and which their all own limits, because now our manufacturing units are complying more stringent units, because most of the export oriented unit are tie up with foreign regulatory bonding like US EPA, ASTM, NIOSH, OROSHA. Most of the ambient air quality covered under the our environmental protection rule EPA, and the NIOSH OSHA will give the work environment, indoor air environment. We will not go through in details in for work environment or indoor environment, but these instruments are the same, will determine and identify the pollutants in the work environment also. Now, water quality assessment, the technique is this GCMSMS technique is widely used to analyze water sample, wastewater sample, and for which pollutant like pesticides, herbicides, and other organics. This information is vital for ensuring safe drinking water and safe disposal or effluent to the different pathways like CETPs, open channel of lake, marine, river, etc. etc. The soil contamination. How GCMSMS is determine the contamination level of soil, sludge, and residue. Likewise, PCB, similar waste, pesticides, and heavy metals. The data helps to assess the environmental risk and guide its remediation efforts also. How to treat, how to treat soil, how to treat water, and how to treat air quality, looking to the concentration of the pollutant. Advantage is always this modern instrument having some advantages, also limitations, also having economical limitations for viability for laboratories like us, because why we use GCMSMS where GC technique is there, but all these instrument having some vital uh, impact on accuracy, detection, and identification. GCMSMS offers several key advantages in environmental analysis. It's highly sensitive, and in ambient air where contamination levels are very less, where dilution is in huge amount and is difficult to identify on routine GC and other techniques. The method is highly specific, enabling accurate identification and quantification of target compound like benzene or other VOCs, having specific library of thousands of ready compound. GCMS or GCMSMS is having inbuilt library. So if we inject unknown sample, it will give, compared to the library, fragment the molecular weight, and it will give the probables. Not 100% sure, but it will give probable compound from unknown sample. So it is very useful where the fingerprint analysis is there. Now I will describe What is fingerprint analysis? Fingerprint analysis, identification of unknown organic waste, chemical, and further identify the liable industry for illegal discharge. If some industry load or discharge their waste, example of solid waste on road, on barren land, or in water bodies, pollution control board will go for the analysis, taking the sample. But how they will identify the industry, unknown industry? Because we all know some of the industry distance from 100 kilometers, and they dump the waste after the distance of 100 kilometers. So it's very difficult, the waste liability and the source of the waste, which industry is liable for this. So this GCMSMS is very vital to identify all the other chemicals of the waste, dump waste, and ultimate identify the source, the manufacturer who dump the waste. So the regulatory body using this instrument very wisely and very widely, and the laboratories like us are also responsible for this identification and ultimate the disposal pathway. Discarded waste on road, barren land, also discharging public sewer, river, marine, and other surface water. GCMSMS technique, it is widely used for unknown sample identification and ultimate confirmation. Limitation, first of all, it's very costly, and the instrument integrity and instrument is very sensitive, so the expertise of the operation is required, and its maintenance is always there. About 20% of the cost is yearly maintenance. So its limitation, looking to its techno-economical, viable, and so accuracy is there. But we should go for this instrument nowadays, looking to its accuracy and widespread acceptability all over the world. Now this is another instrument, the sister instrument of GC, but is very useful for very easy determination of VOC and very easy determination of unknown compound. This is GC headspace or GCMS headspace. This headspace dedicated instrument headspace. So I will describe why the name of headspace. In routine, any type of sample we introduce in GC, GCMS or LCMSMS, it required sample preparation. Air sample will not go directly into the instrument. Water sample, sludge sample will not be introduced directly in this instrument. Middle instrument and sample. In middle, there is some sample preparation techniques, and all the pollutant having some different techniques. So there is no one technique. It's chemical to chemical, molecular weight to molecular weight. Boiling point is also vital for sample preparation technique. So its headspace is one technique. Is very used in pharmaceutical also, and for VOCs which are less boiling point are very easily detected. How very easily detect? I will show you. This GCMS or GC headspace analysis. In headspace, GC or GCMS technique will be employed. VOCs in solid or liquid samples. What we can do? The sample we can take direct sample in 20 ml headspace glass vial. We will put this vial with sample, and the instrument automated heated, and automatically the vapors after heating the sample emit fumes if any pollutants or any volatiles are there. So the fumes of vial vial is 20 ml, sample is 2 ml or 2 gram, the 18 ml of volume covered with the fumes, the air or which emits from the compound inside the vial. So these fumes or gas directly introduced by the system of headspace analyzer to the GC. In GC we can having different peaks, and we will identify the sample contaminants, pollutants very easily. If we didn't have or we do not have some data with the GC, so further we fragment this data to GC MSMS, and ultimate we identify and confirm the unknown compound of the sample. So it is very easy. There is no sample preparation required. So it's easy technique, but it's having its own limitation. Its limitation is there. Only one major limitation is there. It will not identify and fragment higher boiling point component like 2,4-D or pesticides. But our routine pollutants like benzene, toluene, xylene, acetone and other solvents are very easily tested on this headspace analyzer coupled with GC or GCMS, MSMS as per the requirement. Uh, where this headspace is required for our environmental parameters. The matrices can include soil, water, air, and yes, is also covered biological samples also, because some biological samples having some solvents, natural solvents or synthetic solvents. So the samples of these biological parameters which are having very less sample. So the where as and where the sample is very less, so difficult to prepare the sample and inject the different instrument. At that time, if VOC is there, so very small sample require 1 gram, and instrument will hit the sample emit the VOCs, and our GC or GCMS fragment the samples in the different way and identify analyzed, and it will give confirmation. So these VOCs are very easily analyzed on headspace, and nowadays is a compulsion to have such instrument in the laboratory for fast and accurate analysis to comply with latest regulatory norms. I will not go in detail, but this is the use of headspace, wastewater discharge, yes, one landfill gas analysis. So this landfill gas, all we know, the disposal pathway of some solid waste or liquid waste is landfill. Where the landfill site is there, there is some vents. Why they give vents? Because where we landfill the sludge or waste, after some years it will decompose, and after decomposition it will emit the sludge which was in landfill. It will emit some landfill gas like methane, carbon dioxide, and some other gases also, including VOC. So this headspace analysis is very easily uh, technique to determine this landfill gas also. Now soil contamination and soil study, and also air monitoring. Headspace is applied to analyze soil samples for the presence of VOC, which may indicate contamination from spillage, leakage, or other industrial activity. Also, it's easily detect the solvents, VOCs, and from air, stag, and other gas through some other technique also, charcoal or other absorption technique. We will not discuss this technique today, but this technique is very vital, useful for our modern instrumentation. These are just suitable complex. We will not go in detail. Its high sensitivity with trace analysis capability, widely applicable range, and very easily identification of the VOCs. Now, these specialized detectors, which are coupled with gas chromatography, GC, it's ECD, TCD, NPD, FPD, and so others. We will discuss only two, ECD and TCD. This presentation will explore two only essential detectors commonly used in GC, the electron capture detector and the thermal conductivity detector. These detectors offer distinct advantage with analyzing various chemical compound in air, water, and gases. The ECD is highly sensitive detector used in gas chromatography. Nowadays, this detector works by measuring reduction in electrocurrent caused by capture of electrons by electronegative atoms. These are very technical words. We will not go in the detail, or will not go in the detail of the principle of the instrument, but I will highlight this instrument. Why? Because nowadays it is required as per the regulatory norms. So, small laboratory or big laboratory, all these laboratory are and should equip with this instrument. ECD are ideal for detecting halogenated compound such as some organic chlorinated pesticides, DDT, PCB, and other pollutant. They find wide application in environmental monitoring, pharmaceutical analysis, and in food safety testing. I will not go in detail in food safety or food and drug safety, but these all instruments are the similar way of required in such other testing also. Most of halogenated organics are very easily detected by ECD, chlorinated organics, chlorinated solvents, and some pesticides. Chlorobenzene, other solvents which are chlorinated are very easily and very cheaply determined by the ECD. So nowadays most of the laboratories going for this instrumentation, and who are in R&D, they require this instrumentation, and they should know this instrumentation, why this instrumentation is required. PCB in wastewater. Now we are having hazardous waste rule 2016. The used oil and waste oil, it's having PCBs, and there is a regulatory norms of PCB in used oil and waste oil. So we should have this ECD, and we should have the knowledge of this ECD technique, ECD detector with GC and its implication. Uh, Thermal conductivity detector. Similar way, ECD. This thermal conductivity detector is a versatile detector based on the principle of thermal conductivity. It works by measuring the changes in resistance of heated filament caused by the presence of analytic gases. Who are having knowledge of GC or gas chromatography, they know there is some filament, and these all principle correlate with each other. TCD are used to detect a wide range of compound, including hydrocarbons, alcohols, ketone, and aldehydes. They are commonly used in industrial processes for monitoring of gases and environmental parameters. Now ECD are commonly employed to detect chlorinated or halogenated compound in air, water, or any other matrices. TCD plays a crucial role in monitoring industrial emission, ensuring compliance with environmental regulations, and ultimately minimize the pollution if we implement properly. Uh, ECD is high sensitivity detector having its own limitation, and ECD having some radioactive chemicals also. So if we use ECD detector in our laboratory, we should go for the permission of Government of India for this radioactive active chemical, for this departmental permission also. So its limitation for sensitivity, permission from Government of India, and uh, periodically inform the department regarding the ECD testing and others. TCD is widely having more ranges, having strong electron capture properties. So TCD is having very vital role to detect gases like carbon dioxide, carbon monoxide in the air. with very broad spectrum. And today we are having analysis of biogas. This TCD, FID are very vital to use to analyze biogas also for methane, carbon dioxide, carbon monoxide, and other related impurities also. Both ECD and TCD detectors can be used for quantitative analysis, enabling accurate determination of analyte with appropriate calibration standards. Now I will switch over the inorganic part of chemicals. We have discussed GC, GCMS, GCMSMS, and I will discuss after this part HPLC also in summary. But all these instruments related to GC are covered testing of organics, where inorganic part will require further more advanced and other techniques. Inorganic part cover parameters like anion, cation, heavy metals, lead, cadmium, arsenic, mercury, and so on and so. Now, similar way, why we require this advanced and modern technique or modern instrument in our laboratory. ICP, inductive coupled plasma, ICP or ICP-MS, to detect heavy metals. First of all, I given example of heavy metal like lead by spectrometry. It's having own detection limitation in higher ppm, which is not useful because our regulatory limits in very low ppm. So spectrophotometer will not capable to analyze such metal in ppm level. After that, we have atomic absorption spectrophotometer, which is having its all own advantage and disadvantage and limitation of quantify limit in ppm. Now, furthermore, drinking water or groundwater contamination, some ppb level impact of mercury, cadmium, lead will direct impact to human, because its ppb level is toxic considered under the US EPA or considered under the CPCB drinking water guidelines. So, more or less, we should determine the toxic level of these heavy metals or inorganics in ppb or ppt level. Now is the required. Now these days require sophisticated instrument which test in ppb level. So this ICP is one of the instrument which is widely used all over the world for heavy metal analysis. ICP utilizes a high temperature plasma generated by radio frequency waves. We will not go into detail. The plasma ionizes the atoms of the analyte, converting them into the charged ion. These ions are then detected by spectrometer. Application of ICP in analytical monitoring, very wide applications. ICP is a versatile technique employed for analyzing heavy metals and other elements in a wide range of environmental samples, including water, air, and biological tissues also. Example of this advantage and disadvantage: spectrophotometer, atomic, and ultimate we are having now this ICP MSMS technique. So this ICP analyze more than heavy metals in one aspiration, one injection, and it's more than 100 samples together in a very short time, where our spectrophotometer having limitation of 10 to 12 samples per day with interference and all other detection limitations. Atomic absorption spectrophotometer having its own advantage. It will give results of only one element at a time. If we inspire or inject one sample, it will give only one parameter detection, lead or mercury or cadmium, where ICP or ICP MSMS give all the heavy metals together in one injection and hundred of samples at a time in very short period of testing. So its viability, its accuracy, and widely acceptability in the world. We should go for now ICP technique and not atomic absorption spectrophotometer or routine spectrophotometer to determine heavy metals, looking its widespread acceptability and accuracy. This ICP-MS combined the ionization power. I will just like what is ionization and ICP, what is inside. It's having sensitivity of mass spectrophotometer. It separates ion based on their mass charge ratio, allowing for the identification and quantification of individual element. Example like heavy metals like lead, mercury, cadmium, so and so. Same as GCMSMS or GCMS, where organic part will be identified with proper confirmation. Where here ICP require further fragmentation for confirmation. So ICP coupled with mass spectrophotometry will give further fragmentation and confirmation of the inorganics, heavy metals and other pollutants. Water sample is widely used for the monitoring of heavy metals in drinking water, wastewater and surface water bodies like mercury, lead and other heavy metal. Similar way, soil, sludge, residue of the byproduct. Similar way, ICP and ICPMS very easily detect all such heavy metal fastly and accurately. ICP is having high sensitivity, having multiple element analysis at a time. It can simultaneously measure multiple elements in the single sample, making it efficient for analysis for complex matrices also. We will not further discuss which are the complex matrices, but this is the advantage for ICP on atomic absorption spectrophotometer or second instrument of spectrophotometer. So this wide linear dynamic range is concentration. It can be measured accurately within a single analysis and within a fraction of the seconds also. Now IC and chromatography. Separation principle is based on their interaction with ion exchange resin. Why IC is required? We are determining anion, cation by titration sometimes by spectrophotometer, like chloride in water. It's very easily determined by titration. Chloride determination by titration with color changes. But if we are having colored sample, in color sample, our titration will not useful for accurate analysis. If acidic sample is there, chloride is not determined by titration technique or furthermore spectrophotometric technique. At that time, for such samples, because our environmental samples are not compared to drinking water samples or other potable matrices. Most of our samples, most of our matrices are colored and contaminated with other organics also, other chemicals also. So at that time, uh, sulfate, nitrate, chloride will not be determined by routine titration or spectrometry techniques. At that time, IC is one of the only option to detect the same. If we are having acid mixture, having sulfuric acid, hydrochloric acid, nitric acid, how we can identify the percentage of all these individual different acids? So our routine titration technique of acidity, spectrophotometric, colorimetric technique will not be applicable at that time. IC will differentiate all anions, all cations at a time, and it will give the final results of pollutants or elements or chemicals. So it anion and cation are the only instrument on analyze where this hazardous, colored or highly acidic samples or contaminated with organics. IC used to test acid mixture as I told you, and also where sample is colored. Turbid, where colorimetric and spectro technique is not possible. Color mono sample will not be give results of chloride by titration. Why I am repeating? Because I want to give proper understanding for identification of modern techniques, modern instrument compared to our past old instruments. So one another example I will give you where I see and chromatography is useful. Uh, hydrochloric acid, hydrobromic acid, HCl, HBr, or bromine or chlorine fumes from stack gas. So there is no method to detect, absorb these fumes and detect the same by conventional method. No spectrophotometer or routine our method will require or implement or prescribed by international or national organization for test method. So if we want to check hydrochloric acid or chlorine or bromine in stack gas, IC is the only instrument where we can differentiate the parameters like HCl, bromine, others is very easily we can absorb in absorbing material. Gas will go through the absorbing material. Chlorine, bromine and other halogens absorbed in absorbing media, and after extracting this absorbing media we can inject in IC. So whole different spectra, whole different peaks of chlorine, bromine nitrate, sulfate for sulfuric acid emission, uh, other acid gas emission very easily determined by this IC. So, advantages of this ion chromatography: high sensitivity and very easy identification with limited, very limited interferences compared to other high instrument like GC, GCMS, LCMS. Now once again, we switch over the part on organic, inorganic, and now once again organic part. Most of are knowing HPLC, high performance liquid chromatography. HPL separates compound based on their polarity. We will slightly or 2 minutes discuss the principle and techniques. So we will understand further where when we will discuss the HPLC. Okay, the stationary phase is a solid material with different polarities, and the mobile phase is a liquid solvent. The compounds are then evaluated from column in order of increasing polarity, and they are detected by detector that measure the concentration of each compound. The mobile phase, or we can say the column inside the HPLC, we can insert the sample, prepared sample. The compound in the sample interact with the stationary phase of the column based on their polarity. The more similar the polarity of the compound is to the stationary phase, the longer it will retain in the column. So it is the technicality of the instrument, detector, column, stationary phase, temperature controller, injection, and others. We will not much go into detail. Organic pollutants, widely used to analyze organic pollutant in water, soil, air, including pesticides, industrial chemical, and pharmaceuticals. Once again, I will tell you liquid chromatography like HPLC or gas chromatography. Only two difference between this implication or usefulness. Higher boiling point component will not be easily analyzed by GC technique, as well as lower boiling point component will not be tested or analyzed by this liquid chromatography or say HPLC or say LC-MS. So this HPLC technique is useful for higher boiling point component, which is very difficult to analyze by GC or GC-MS. 2,4-D or most of the pesticides are having higher boiling points. Most of the pharmaceutical component, pharmaceutical intermediates are having higher boiling points. So HPLC is widely used in pharmaceutical companies. One example, if we go to the big pharmaceutical factory, they are having more than 50 HPLC instrument in a line. So the application of this HPLC is widely used for pharmaceuticals and intermediates, but these pharmaceuticals residue can be very easily determined from water, wastewater, soil, as well as air by this instrument. HPLC helps to determine pesticides residue in water and soil, ensuring food safety and protecting agriculture environment, but its limitation of detection level is much higher compared to HPLC MSMS, which we already discussed in the first slide. So it's having some different type of detector. It's UV visible detector, fluorescent detector. We will not go into detail, but we should know the HPLC having column inside, having one oven inside, having some detectors for detection. Now, one more vital instrument nowadays, total organic carbon or total nitrogen analyzer or TOC analyzer. Okay, this TOC analyzer is very much required nowadays. How? Why it's required? One simple example is that uh, nowadays all and most of the CTPs are equipped with TOC analyzer. Why? COD analyzer is there. So, what is the requirement of TOC analyzer? COD covered all organics, TOC covered all organics. So, why we require such costly instrument where COD is there? Okay, so COD is a very easy technique, very cheap technique to identify organic load, but it's having its own limitation. Most of are knowing if you are in the laboratory field or in the research field or in student where when we you tested some samples of COD in your laboratory, in college laboratory, higher halogenated compound, chlorine, bromine are false positive to the COD. If organic zero, organic load will be zero, but it will give thousands of result milligram per liter of COD. So it's false positive if there is halogenated compounds are there. Nowadays, most of the effluent, most of our waste contain halogenated compounds like PCB, organic halogenated compound, pesticides, and our highly colored sample, highly acidic sample compared with organics or saline water. It is very difficult to test COD in this sample. So nowadays regulatory body is also preparing some draft to replace COD with TOC. It is prepared long 10 years back, but looking to the cost of the instrument and availability of the instrument, today still is in the draft, but uh, other countries like USA or other developed countries, COD replaced the TOC. So TOC analyzer works by oxidizing organic compound to carbon dioxide for TOC analysis. We will not go into detail. The
Shivan
oxidation
sir,
process.
sorry to interrupt.
Yes, sir. Please.
Yes, we are having 5 to 10 minutes more.
Okay, sir.
We shall try to conclude the important slides,
Okay.
and
So I
after that we can spare 5 minutes for question answers.
okay. So I will
Devang bhai,
highlight.
actually, please take 5 minutes only because
Okay.
another speaker is
Yes,
from
sir.
Canada
Definitely.
and
Yes, sir.
she has some time constraint.
Yes, sir. Definitely. Definitely. I will conclude within 5 minutes, sir.
Yeah.
Okay, sir.
Thank you.
Okay. Thank you. So we'll not go into detail. This TOC analyzer is widely applicable and widely used up country also, and nowadays in India this TOC analyzer is a must to detect TOC level in the water, sludge, or any byproduct of acid, byproduct of organics where TOC analysis is required. So this is precision accuracy. Same way, CHNO elemental analyzer is there. So this latest instrument, this modern instrument is primarily requirement nowadays for accurate analysis. Now, HPLC techniques, instrument analysis techniques, same way sample preparation techniques, will not go into detail, but this is some technique for using instrument very accurately. Now one instrument is remaining. We all know dioxin furan. There is only one instrument which detect dioxin furan from gases or from waste. Very few laboratories in India. I think one or two laboratories in India having HRMS. We are also not having and sending the samples to the upcountry. But why we should know regarding the HRMS also? Because this is a higher end instrument, and now it is the answer after 10 years in India. We should go for HRMS. Now one more instrument, sir. 1 minute, please. VOC detector. We are having VOC analyzer, VOC instrument, principal by PID. It will give immediate answer of the VOC's presence in the air. So it is very easy to carry, and it's remove all the interferences where in past we sample or absorb in some absorbing media, extract it and then inject it in the GCMS for determination of VOC. Now we are having this very widely used acceptable VOC meter. Will not go into details, sir. Same way, the online gas analyzers for SO2, NOx, HCl is widely used nowadays. Same way, in our ambient air quality guideline, carbon monoxide analyzer of NDIR principle. So it is a compulsory equipment to detect CO from ambient air as per the ambient air monitoring guidelines of CPCB or NIF. Now we conclude. Here we develop into the world of advanced instrumentation for environmental analysis. We explored how these instruments play a vital role in monitoring and understanding our environment, and enabling us informed decision for a sustainable future. Air quality, water quality, waste analysis, all are primarily required these basic or high-end instrument nowadays. So, I have concluded my presentation. Hope all you enjoyed. Some technical words are there, some highly technical words are there. Sorry for that. But I should include all these words and phrases also. Thanks a lot, all of you, for listening in
Thank you, sir.
very patiently. Thank
Thank
you.
you, sir. Thank you, sir, for this very informative session. Dear participants, those who have written their questions in the chat box, I have unmuted them. You may please ask the questions. We are running
Yeah, can I
short of
ask?
time.
Yeah,
Yes, yes,
just a
please.
small
We
question.
have only 5 minutes.
Yes,
Yeah, just a
please.
small question.
You
Yes,
may.
Yeah, is this radioactive permission is to be taken from BARC, sir?
yes, sir, from BRC.
Okay.
And
And I was
we should file a periodical inventory also and
yes.
periodical information also 6 monthly to BRC.
Okay. Yeah, that's what we do in industry also
Yes,
for
sir.
the for the plant scales.
Yes, sir.
Thank you.
Thank you.
Anybody is having any questions?
Hello, sir.
Sir, may
Am I
I ask?
audible?
Yes, yes, you are audible.
Sir, my question is if we testing pesticide sample for particularly wastewater and soil, right? Then which detectors we may use?
Now there are numbers of detectors compared and required as per the detection limit. But in layman language, nowadays pesticides will not be detected very easily or accurately with GC or any other detector. Now GC-MSMS or LC-MSMS is the only option to detect accurately and compare with our regulatory norms.
Okay, sir. Thank you.
Sir, may I ask?
Sir, may I ask a question, please?
Yes, please go ahead.
Uh, Hello, sir.
Please
I,
start
w-
your
I
camera.
w-
Yeah.
Those who want to ask the question are requested to on their cameras. Yes, please go ahead. Somebody was willing to ask a question.
Hello, sir.
Yes, please.
Sir, I have a question regarding GCMS. If our interested, if we have two components that uh, those are interested to find out, and suppose their boiling point is same, then which one will elute first in system?
As it's logical. Lower boiling point first.
Okay, those who are having lower boiling points will elute
Yes.
first. Okay,
Yes.
sir. Okay. Okay.
Okay, this will be the last question. Hitesh Soni,
Sir,
Hello,
please go ahead. Yes,
uh, hello. Hear
yes,
me,
yes,
sir?
you are audible.
Yes, you are audible.
Sir, just I have only one question. Just organochlorine and organophosphorus, both parameter can be do in analysis of FID detector.
No. Organochlorine required ECD detector
Yeah.
and phosphorus require NPD or FPD detector. But
Uh,
if
okay.
there is
NPD and FPD.
GCMS, if there is GCMS, no other detector required.
Hello. Okay, sir.
Okay, thank you so much, sir, Devang sir, for your valuable time and knowledge
Thank you.
sharing session. Sorry, we had to interrupt you in between, but our next speaker is already in the lobby and she has some time constraints. That's the reason. We will arrange your session again in future. Thank you so much, sir.
Thank you, Devang bhai.
Thank you, Dr. sir. Thank you, Hiren sir. And thanks all
Thank
of you.
you. Okay, sir. You may stop sharing your screen, sir. I will just take a moment.